Hi, good evening, everyone. Good evening. So, how are you? Okay. What are the gossips, or we can say chat? I can see the chat that is related to IPL. One IPL season is also going on, but at the same time, you also have to focus on little bit on what that is your subject, because it will. Good evening, everyone. So obviously you are enjoying your home food, because after 1 month you don't have the home food, you are away from the home, but no doubt that some food is there. Good evening. So good evening. So anyway, in the last class, we were talking something about what? Good evening, Google Pixel 9, good evening, because your name is not there. Yes. So in the last class, we were talking about what? We are talking about the cell theory. So what does it mean by theory? So theory, we can say that experimentally proven data. Experimentally proven data is known as what? Theory. But whatever the things that is going in my mind, it's not theory. Once I prove it experimentally, then I can call it as a what? It's a theory, and this theory I can publish to everyone. Like, because personally, if I am a scientist, or we can say if you are a scientist, you can't go individually door to door and will say that I have experimented on cell, or I have observed like a plant cell is having outermost boundary is the cell wall, and animal cell is having the cell membrane. You can't go individually to each and every person. So that's why they will publish in the public, so that everyone can read, or we can say everyone can access this. Okay. So anyway, cell theory, first question is the two German who gave cell theory. The name of that two person was, like one is, I'm just waiting for the answer. One is Swan and other one is Sladen. Okay. Next one is after that one more scientist, again German comes, and who has rectified the observations of what? Sladen and Swan. So what's the name of that person, that fellow? Rudolph. Good. After that, combined, all three members come together and they have said that related to the cell, these are the things should be there. And one more thing, I can say virus. So we have studied a little bit about the virus. So cell theory is applicable to virus. True or false? Cell theory is applicable to virus. True or false? Yes, many of the students is Nagarjuna, Regina. So obviously it's a false. It is not applicable. It is not applicable. Why? Because virus is considered to be acellular. Virus is considered to be acellular, means it doesn't have any kind of cell membrane. Metabolism is there, but this metabolism dependent on what? Dependent on the host. Like, let's say I'm getting infected by some virus. So I will do all the metabolism for the virus. Virus itself is not doing anything. So virus is enjoying in my body, nothing else. Okay, so that's why I can consider is cell theory is not applicable to the virus. Okay. After that, what else? Next is prokaryotic cells and eukaryotic cells. I made some differences. Okay, so a lot more differences was there that we have discussed in the last class, but main difference is one is having the nuclear membrane, that is eukaryotic cell. Other one is membraneless, that is prokaryotic. One is having the double-stranded but circular DNA, that is prokaryotic. One is having double-stranded but it's a linear, that is known as eukaryotic. Membrane-bound organelles are present in the eukaryotes while it is absent in the prokaryotes. Histone protein present in the eukaryote but absent in the. So these are the few things that we have discussed in the last class. But today's topic is nothing but the very interesting topic, and we can say it's a prokaryotes. Prokaryotes, obviously, it looks simple, but actually they are not simple. Once it comes to the behavior, it is little bit, or we can say it's a more than imagination. Like its complexity goes our imagination. We can't even think. We can't even think. Okay. So here it's a. Today we'll go with the another topic that is nothing but the prokaryotic cells, or we can say prokaryotes. Okay. So in the prokaryotes, firstly, we have introduced little bit about the prokaryotes. Okay. But again, what are the organisms comes under the prokaryotes? I will just write the few names of that. Mainly, we can say most of all the prokaryotes, prokaryotes belongs to what? All the prokaryotes grouped together in the kingdom, the name of the kingdom is nothing but the monera. So monera is nothing but the name of the kingdom, and whoever belongs to that is nothing but that it's a monerans. Can we say that we are Indians? Because we belong to the country that is India. So that's why we can call ourselves as an Indian. Same as it is, prokaryotes belongs to what? Kingdom monera. So all the members of this kingdom monera, I can call it as a monerans. Okay. But in that case, we can say the most important members of this kingdom monerans. So that I can call it as a bacteria. So bacteria, we can say it's a whole and sole member of this kingdom, that is the kingdom monerans. So in this case, we can say sole member of the kingdom monera is what? Is bacteria. Is bacteria. Okay. So this is the one part. After that, if you are moving to the next, so what other members are there? For example, I can write few members. So mainly it's bacteria, or it's better to say it's a eubacteria. It's a eubacteria, and in that case, the eubacteria, here you can find that is nothing but blue-green algae. That is a blue-green algae. After that, now we'll move to the next. Again, it is also having another member that is the most primitive member, that is known as archaebacteria. And archaebacteria is considered to be as a what? Archaebacteria considered to be as the most primitive, or we can say the eldest member of the kingdom monerans. Okay. Now, again, a few more members are there, they are kind of exceptional. Why? Because it is not following all the behavior of what? Kingdom monerans. Okay, no doubt they are prokaryotes, but still they are not following. So I can call it as a what? mycoplasma. Mycoplasma. Okay. So next one is, in this mycoplasma, again you can find one is nothing but piplo, or we can say it's a pleuro. Pleuro means it's a fluid. Okay. So pleuropneumonia like organism. Pleuropneumonia like organism, or in the abbreviated form, I can call it as a what? It's a piplo. I can call it as a piplo. So it's nothing but a locase monera and protista. Both are nothing but unicellular. No doubt, one is eukaryotes and other one is prokaryote, but both are nothing but unicellular. Okay. After that, if you are moving to the next, where it is, or we can say where it stays. So I can say it's nothing but the most diverse. It's a nothing but the diverse, most diverse. So wherever you can imagine, it is present. In the soil, in the water, in the air. So everywhere it is present. Everywhere it is present. Even in the heart's condition also it is present. So if you are talking about the habitat, you can find that here it can also be present in the soil. Next one is, it is also present in the water. Next one is, it can also be present in, it can also live inside some other organisms, or we can say it's a host. Means it can also be the parasites. And this parasites, we can say, so this parasites, so what would be the host? Host can be plants, or animals, or any other. It can act as a, can be the host. Sometimes it is present in symbiotic relationship. Symbiotic relationship means what? Like one organism comes in contact with the other organism. After that, they are planning like, I will do some of the task, you do some of the task, and we will live happily, we will live comfortably. Okay, so that is nothing but it's a symbiont. So some are present in the form of symbiotic relationship. So these are nothing but the habitat. A lot more habitats can also be there. Like if you are talking about the archaebacteria, so archaebacteria is more diverse, or we can say it can be present in the more extreme condition. So what can be the extreme condition? More salty water, more acidic water, more warm water, or we can say high temperature. So these are nothing but the few areas, or we can say few habitat that is extreme for us, but for them it's a very easy, very comfort life there. Okay, so these are the few introduction that is related to that. Okay, now I will move to the next part, that is the structure of what? Structure of a typical prokaryotes. Structure of typical prokaryotes. That mechanisms and all I won't go in detail. So firstly I will go in detail with the what? That is a prokaryotic structure. How it looks like, or what are the structures are present, and which structure helps in performing what type of we can say functions. So now I will draw one very simple diagram, because the bacteria looks very simple, but when it comes to the behavior, it becomes so complex. Then it's very difficult. Yes, the members of monerans, like one question is there, like the members of monerans all are prokaryotes. Okay. Now, if we are moving to the structure of what? Structure of the prokaryotic cells. or we can say structure of typical bacteria. Structure of typical bacteria. A typical bacteria means what? It's a eubacteria, or we can say it's a true bacteria. Eubacteria, or we can say it's a true bacteria. Okay. So just imagine one bacteria is there in your bedroom, or we can say wherever you are staying or wherever you are sitting, there is a bacteria. So everyone got the bacteria nearby your place. Okay, so just imagine how the bacteria looks like. Okay, so I am like here also one bacteria is there. I'm just imagining how this bacteria looks like. How this bacteria looks like. But obviously it's a microscopic structure, so we need a microscope to properly visualize the structure of the bacteria. Okay, you can't see. Obviously, once I will draw, it will be available on the board. So I'm just going from outside to inside. Let's say I have one bacteria, and once I see this bacteria, I can find the outermost layer of bacteria, and that outermost bacteria or bacterial layer is known as glycocalyx. That is very important for the bacteria. So the outermost layer of the bacteria is nothing but the glycocalyx. So let's say I am drawing a bacterial structure. I'm drawing a bacterial structure, and the outermost layer of this bacterial structure is nothing but the glycocalyx. And this glycocalyx may be present in the form of hard capsule, or it can be present in the very slimy, very thin layer, and that thin layer is known as slime layer. So it can be present in both the forms. So this is the first layer, or we can say it's outermost layer. So it's better to write here, or we can say level here. This part is nothing but the. So what's that? This is known as glycocalyx. That is the outermost layer. Now, once I enter into the bacteria, once I am seeing the bacteria inside. So after that, which layer comes? Now I find one more layer, but this time again the layer is dead. So this glycocalyx is also dead. Inside that, one more layer will come, that is also dead. And this dead layer is known as the cell wall. It's nothing but the cell wall. Cell wall will be the another layer. So it's nothing but the cell wall. So the second layer is nothing but the cell wall. Again, I level the diagram here itself, because it's very easy to correlate. It's nothing but the cell wall. So just inside the glycocalyx, what is there? Cell wall. So outermost layer is glycocalyx. Inside that, it's a cell wall. Okay. So all moneras are double membrane. Double membrane we can't say, but boundary is there. More than one boundary is there. Okay. So this is the second layer. Now, am I audible to everyone? Like one student saying like I'm not audible. If it is audible, just thumbs up. Okay, yeah, it's reachable. So in this case, now I will move to the next one. So what's next? Next, I can find it's a very thin, very delicate, very flexible. It can change its shape. So that I can call it as a what? Cell membrane, or in another way we can say it's a plasma membrane. Okay. So how many of you in this class having the cell membrane? Everyone is having the cell membrane? I hope answer will be yes, because we are animal cells. We are having the animal cells, and the outermost boundary of the animal cell is nothing but the cell membrane. That is a highly thin, delicate, flexible structure. And cause of this cell membrane, I am able to write. I am easily able to move from one place to another place. Why? Because I am having the cell membrane, not cell wall. If it is a cell wall, for me it's very difficult to write, or we can say very difficult to speak. Okay, so that's the advantage of being the animal cell, or we can say only cell membrane is there. Okay, now it should be in hexagon. No, there is no such kind of thala, like thala dhoni 7, like there is no hexagon kind of structure is there. So simply I am just making one diagram where I am able to level all that part. Now the innermost part, but this is living. This is the living structure, and in that living structure, so this is nothing but the living structure. And this living structure, so this is nothing but the living part. This is nothing but the living part. Here it's okay. So this is nothing but the cell membrane. No doubt, no, it's not villi locus. Villi we can't say, but the cell membrane itself modify themselves to perform different type of functions. Different type of functions. Why? Because it's a unicellular, and unicellular is having only one cell, and that one cell has to perform everything. So that's why the cell membrane itself modified into the n number of structures to perform different functions. Okay. Now again I just try to level the diagram. So here this part, the folded part of the cell membrane. So firstly I will add this point, that is this part is nothing but that this time it's a cell. So what's that? It's nothing but the cell membrane. Sir, cell wall made up of peptide. Yes, it's a peptidoglycan, but that we will go later. So once I draw the diagram, thereafter we'll go in deep, or we can say the descriptive part of that. Okay, so now it's a cell membrane, but sometime I am finding the cell membrane is folded. So this folded part of the cell membrane, I can call it as a, so what's that? Can you read this? Yes, that is mesosomes. So mesosome is nothing but the infolding, or we can say some type of folded forms are there. And when the folding is required, so the folding is required whenever if you want to increase the surface area. So basically folding will increase your surface area, nothing else. Okay. After that, now we'll move to the next structure. This structure is somehow I have drawn here, but its a place can be variable. And this I can call it as a chromatophore. But this chromatophore is not present in all bacteria, because the bacteria which is autotrophic in nature, or we can say photosynthetic in nature, will have this chromatophore. Not all bacteria is having the chromatophore. So I am just drawing the typical bacteria. Okay. After that, now we'll move to the another structure. So this structure I can call it as, so what's that? It's a fimbriae. It's a fimbriae. After that, one more structure is there. This I can call it as a pili. So this is nothing but the pili. Now next one is, what happens if cell membrane is not present? So if cell membrane is not present, it's a very difficult to have some structure, to have some organized structure. If you don't have organized structure, then obviously you won't get the shape. You won't get the shape. What is meant by lysosome? So lysosome is membrane bound structure, and here it's a different. Mesosome is different. Okay. Now we'll move to the next part. So next part is this. This is nothing but the flagella, which helps in the movement, or we can say which helps in what? Locomotion from one place to another place. Or let's say one bacteria is there in Andhra Pradesh, and bacteria this time wants to go where? Wants to go to Telangana or some other place. How it will go? So it will go with the flagella. Okay. Now after that, if you are moving to the next part. So this membrane, inside the membrane, we can found the ground substance. We can found the ground substance, and that ground substance I can call it as a cytoplasm. Ground substance I can call it as a cytoplasm, and cytoplasm is the general structure and which you find everywhere. Like if it is a live cells, either it's a prokaryotic cells or eukaryotic cells, this is the common structure. What's that? That is a cytoplasm. Okay. So this part is a cytoplasm. And actually cytoplasm is nothing but it's a kind of ground substance where all the other structures are embedded, all other structures are floating. Okay. So how many of you have seen the pond, or we can say lake? Everyone has seen, right? The pond and lake. Let's say I'm just considering one pond is there. So that pond, can I call it as a cytoplasm? And whenever you are swimming in that pond, so that swimming part, we can say different structures like ribosomes, or in other word we can say chromosomes. Okay, so is that clear? So it's a cytoplasm. Inside the cytoplasm we'll go one by one. What are the structures? The first and the most important structure of any cell is it should have the genetic material. Without genetic material, it's a very impossible to imagine the life. So here I can draw one genetic material. Earlier I have said that this is double-stranded. So how many strands I have made? I have made only one strand. So I just go to another strands, like this is the second strand. This is another the second strand. So can I call it as a double-stranded? But how it looks like, circular or linear? So obviously I think your answer should be the circular. Okay. So here one more thing I can add is double stranded, but it is circular DNA. Double stranded circular DNA. Okay. So now next one is what? What else is present inside that? So I found one more structure that actually helps in what? Protein synthesis. Means obviously, Dakshita, yes, it's nothing but the ribosomes. Protein synthesis means it's a ribosomes, and yesterday itself we were discussing about the ribosomes. What type of ribosomes? 70S or 80S? Yes, locates. It's a 70S. 70S type of ribosomes will be there because this is the characteristic feature of what? Characteristic feature of the prokaryotic cells. You don't find 80S type of ribosome in this case. Okay. Now, so 2 structure is done. After that, now we'll move to the another structure. So another structure you can find the stored food. So sometime you can find the stored food inside that. So let's say these are the stored food. These are the stored food I made. Actually the stored food as well as, so this stored food I can keep it inside one body that is known as inclusion body. Inclusion body. Okay. Next one is a inclusion body also have one more part that is nothing but the what? It's a gas vacuole. So gas vacuoles you can find inside what? Inside the prokaryotic cells. Prokaryotic cells. So here another structure that I can call it as a gas vacuoles. Okay. Now here what I have made one structure. If I have made one structure, so this structure is related to what? Related to the pigments. But not all bacteria is having the pigments. Why? Not all bacteria is performing photosynthesis. Most of the bacteria are heterotroph. So if it is a heterotroph means I will depend on some others for my food. So same as it is, most of the bacteria depends on some other for their food. Only few bacteria that can synthesize its own food. Okay. So here it's I will add some pigments. Here I will add few pigments. And this pigment is attached to what? Or we can say it is related to the chromatophore. So here we can say it's a pigments. Pigments are present. Okay, so this is nothing but the actual structure of what? Actual structure of bacteria. Bacteria is not looking 100% similar to that. I just try to draw different parts of the bacteria. Okay. So if you want to draw, then just draw this diagram. Sir, where is nucleolus? Infinite, there is no nucleolus present here. There is no nucleolus present here. Why? Nucleolus is present inside the nucleus, and nucleus is actually not present here, or we can say nuclear membrane like structure is not there. Okay. So next one is it's a structurally very simple and metabolically it's very complex, because you can find a lot more, or we can say different type of eating process. It can be present in the holozoic form, so means parasitic forms. It can be the saprophytes. It can be the autotrophs. So different methods of getting the food will be there. So that's why I can call it as a complex, or we can say diverse metabolic. So metabolic diversity will be there in the kingdom monerans. Difference between the, see, firstly draw all the structures, thereafter we'll go one by one. Okay. So now I will just, you can I will say what that is a QR code. From there you can write. Okay. Nucleoid and rest of the part I will do. So we'll go one by one. So the first structure, like once you come across, you are coming across one bacteria. I come across one bacteria. I find that, oh, this bacteria is having very tough glycocalyx. And if it is a tough, I can call it as a capsule. So if I'm talking about the structures, we'll go one by one. Structure, the first structure that will be present here. It's a what's that? It's a glycocalyx. And this glycocalyx, its composition is little bit different. Its composition is little bit different. Sometime I can call it as a capsule, other time I can call it as a slime layer. It's a very sticky in nature, or we can say so very watery, or we can say fluid in nature. So here the first part is outermost, and if you are talking about the compositions, so compositions generally composition is generally having what? Polysaccharides. Composition, it is having the polysaccharides. Polysaccharides plus sometime you can find amino acids, or we can say it's a protein. Composition is polysaccharides, and obviously you will ask what is polysaccharide. So polysaccharide is nothing but the poly means what? More than one. More than of what? So here we can say more than one saccharide. More than one saccharide, or we can say more than one monosaccharide is known as what? Polysaccharide. So here we can say plus sometime, like not all the time, sometime amino acid can also be there. Amino acid can also be there. Okay, so that's why I can say that it's a forms or types. One is known as capsule, and other one is nothing but it's a slime layer. Other one is nothing but slime layer. So some of the bacteria is having the capsule, other bacteria will have this slime layer. That depends on the genetics, that depends on the whatever the information is stored in its genetic materials. Okay, so this is the first part. Okay, so what is mean by polysaccharide? Polysaccharide is nothing but the more than one monosaccharide. Like glucose is a monosaccharide. So if glucose will repeat itself multiple times, you will get polysaccharides. Okay, so here we'll move to the next one. So this is the first structure, that is a glycocalyx. Now I will go to another structure. Next structure is nothing but the cell wall. Next structure is nothing but the cell wall. Can you tell me it's a dead or living? It's a dead or living. So obviously, yes, Gana, the answer is dead. Kavya. So here it's a structure is dead. And if something is dead, then obviously sensitiveness is very less. So that's why I can call it as a what? Rigid structure. Dead, rigid structure. So if it is a dead and rigid, means it provides some kind of strength. Nostoc is unicellular because again it belongs to what? It belongs to the kingdom monerans. So dead, rigid structure. And if you are talking about the compositions or components of the cell wall. So no, it's not cellulose. Glycocalyx is dead. So glycocalyx is dead. This one is also dead. So composition is peptido. So once I write peptido means what? Peptido means it's a peptide, it's a protein. Peptido means it's a protein related things are there. Next one is a glycan. So glycan means what? Carbohydrate is there. So carbohydrate plus protein both can be there, or we can say it's a component will be there. So that's why I can call it as a peptidoglycan. What's that? It's a peptidoglycan. After that, now we'll move to the next part. So in this case, based on the cell wall, I am not going in much deep, but I will just give you an idea. So based on the cell wall, bacteria can be of two types. One is gram-positive bacteria, other one is gram-negative bacteria. Yes, good, Dikshita. So it's a gram-positive and gram-negative bacteria. So why it is gram-positive and why it is gram-negative? So if I am saying like if any bacteria is following the rules of Gram was a scientist actually. Gram was a scientist who has done some staining, or we get some coloring of what? Coloring of the bacteria. So whichever the bacteria is following the Gram's rule is known as gram positive, and other one which is not following, it's a gram negative. Okay. So is that clear? Like what is gram positive and what is gram negative? So the bacteria which is following gram is known as gram positive, and other one is not following, means it's a gram negative. So mainly two type of bacteria will be there. So one is gram positive bacteria, and another one is gram negative bacteria, because gram likes only the colorful part. So which one is colorful? Obviously positive, gram positive will be the colorful part. So here it's nothing but the colorful, and while in this case this is nothing but the colorless. So according to the gram, like whoever retain the color is gram positive, and who lose the color is known as gram negative. So this is nothing but the cell wall, composition of the cell wall will be slightly different in the gram positive bacteria as well as gram negative bacteria. Okay. So is that clear? Now we'll move to the next. So this is related to somehow cell wall. After the cell wall, now we'll move to the another structure. That another structure is nothing but the cell membrane. Yes, cell envelope will come later. So the next layer is nothing but the cell membrane. So everyone is having the cell membrane here. Same as it is, bacteria is also having the cell membrane. Bacteria is also having the cell membrane. So if you are talking about the cell membrane, like I will add a little bit few points. So first point is what? It's a living structure. It's a living structure. After that, next point is it's selectively permeable or semi-permeable. So one point is there, selectively permeable means what? Like only few molecules is allowed to pass through it. Means not all the molecules are allowed to pass through it. It's like a selection process. Like in the NEET, not all students are allowed to go to the medical college. Same as, so that's why, can we say it's a selection process? So here also it's a selection process is there. So it's a living as well as what is the type of permeability? So it's a selectively permeable. It's a selectively permeable. It's a thin or thick structure. So obviously it's a thin and flexible. Thin and flexible. So why it's a thin and flexible? Flexibility is required. If it is not flexible, not a single thing will enter into it. So that's why it should be what? It should be flexible. It should be flexible. Okay. So it's nothing cell membrane. Cell membrane is the living structure. Selectively permeable means it allows very special person, very special molecules to cross through that. Okay, it's nothing but thin and flexible. If it is thick, no one can enter into it. So that's why thinning is required. Now, next one is the structures. So the structures here, which is related to what? Which is related to the cell membrane. I will go one by one. That is the structure which is related to what? Which is related to the cell membrane. So the first structure that we were discussing, that is the mesosomes. So what is the name of the structures? Mesosomes. So mesosomes means, can we say it's like infolding is there, and that infolding will increase the surface area. So infolding is inside the cytoplasm or outside the cytoplasm? So obviously my answer is inside the cytoplasm. So here we can say one point I can add, infolding towards cytoplasm, or we can say infolding in cytoplasm. That's the one. What are the uses of that? Why the bacteria is trying to do this? So bacteria will do this because more surface area is required. So firstly I can add is increase in surface area. Increase in the surface area. Now you can ask one question, what is the advantage of increasing the surface area? So if I increase the surface area, can we say that I can keep more number of structure in that area? So if I want to keep more structures at the same time, at the same place, I need to increase what? I need to increase the surface area. Okay. So here it's an increase in the surface area. After that, what else the function related to that? One, we can say it is also related to the respiration, or we can say oxidation of food. The enzyme that is related to oxidation will also present here what? Where? It's a mesosomes. But do we have mesosomes? Can you answer like, do we have mesosomes, or we can say, do you have the mesosome? Yes, Regina, it's a no. My answer is no. Why? Because we are having the mitochondria. Very organized structure where each and every compartment is performing a specific type of oxidation, or we can say a specific type of energy generation method is there. So that's why we don't need mesosomes. Already we are having what? We are having the mitochondria. So the one who is not having the mitochondria can have this mesosomes. That is a temporary structure, we can say. Temporary structures, we can say. So increase in the surface area. So now we'll move to the next. It is related, means we can say enzymes present here, that is related to what? That is related to oxidation of food. That is related to oxidation of food. And which process helps you to oxidize? Which process is helpful to oxidize your food? So can we call it as a respiration? Yes, that is respiration. So here it's a Good, some of the students are answering properly. So it's very impressive. Now, after that, we'll move to the next one. So in the next one, what I can add few more points. So sometime what happens? Let's say this is the mesosome. This part is the mesosome part, and related to the mesosome, sometime the DNA is attached. Sometime DNA is attached. So if DNA is attached, means it is also related to the DNA replication. It is also related to the DNA replication. So here we can say DNA replication can also be there. Or we can say, so DNA replication is also there, that is related to mesosomes. Now, after that, what else? Sometime this mesosomes also helps to form the cell wall. Also helps to form the cell wall. So here also I can add that is a cell wall. What? It's nothing but the cell wall secretion, or we can say cell wall component is synthesized from that part. Cell wall secretion is there. DNA replication, yes. What is the difference between DNA replication and DNA distribution? So replication means what? You are making one more copy. That is the replication. And distribution means that same DNA, whatever you have synthesized, is present at some places. So, or we can say it's distributed among the daughter cell. So firstly you have to replicate, then you can distribute among the daughter cell. Okay, so is that clear? Now we'll move to the next. So one part is done, like mesosomes. How cell membrane contribute to cell signaling? So obviously cell signaling means what? Some sensitive part will be there. Sensitive part will be there, and this sensitive part means at least it should be living. It should be living. So that's why it's a cell membrane, and in the cell membrane many enzymes are there which can receive the message, or we can say which can transfer the message. So that's why it is also helpful in signaling this. So is bacteria containing germ layers? What does it mean by germ layer? Bacteria containing germ layers. So anyway, like if you are moving to the next, mesosome is done. After the mesosomes, what else? The structure related to the cell membrane. I think second structure which I have drawn there is chromatophore. Chromatophore. So can we say that this chromatophore is nothing but the related to the pigments, or we can say pigment related structure. Pigment related structure. So if it is a pigment related structure, where it should be present in autotrophic bacteria or heterotrophic bacteria? So obviously your answer should be autotrophic. Okay, but in that also autotrophic, it is having some kind of different techniques. One is chemoautotrophs, and one is photoautotrophs. So photo will have this, chemo doesn't require. Chemo, like it will take the chemical, or we can say take the energy from the chemical substance. It will take the energy from chemical substance, means that doesn't require. So it's a pigment related structure. Next one is, after that we'll move to the next part. What else was there? So another structure was there, that is nothing but the pili. So pili is also the extension of what? Extension of what? Extension of the cell membrane. Extension of the cell membrane. So it helps in what? It helps in reproduction, or we can say it helps in reproduction, and that too it's a special type of reproduction. Like normally it follows what type of reproduction? It's a binary fission. It's a very simple, very normal type of reproduction. But one reproduction is there, that is a conjugation. That is conjugation. Conjugation is a type of special case where the genetic material is transferred from one bacteria to the another bacteria. Sir, is bacteria no, there is no diploblastic and triploblastic. For that, the organism should be what? Organism should be multicellular. Okay, infinite. So it's a pili. Means it will form what? It forms what? Conjugation tube. It forms what? Conjugation tube. And through that conjugation tube, the genetic material will be transferred from one bacteria to another bacteria. Okay. So here it's a, now another structure, it's a fimbriae. It's a fimbriae was there. So actually it's a fimbriae is nothing but the very simple hairy-like structures you can find. Hairy-like structures you can find. How do chromatophore contribute color changes? Bhavna. So actually chromatophore is a structure that is, or we can say upon which many pigments are there, or we can say colorful pigments are there. So that's why chromatophore looks colorful. Okay. So here it's a fimbriae. Fimbriae, what it does, it's a brush-like structure, or we can say it's a hairy-like structure. Hairy-like structure, and this hairy-like structure is like, let's say this is the bacteria, and upon this bacteria, this is nothing but the fimbriae. This is nothing but the fimbriae, and this fimbriae will try to hold this substratum, or we can say will try to hold this surface. Try to hold this surface. So helps in attachment to what? Helps in attachment to the surface. Surface can be anything. It may be your body, it may be my body, it may be the board, it may be the soil, it may be rock, it can be anything. It can be anything. So this is another fimbriae. This is another fimbriae. So after the fimbriae, what and the last structure that is related to the cell membrane, is the flagella. Flagella is the last structure. So flagella is nothing but this is the structure which helps in what? Locomoting from one place to another place, or we can say it also helps in movement. It also helps in movement. And obviously I can't say that all the bacteria is motile, all the bacteria is moving from one place to another place, but the bacteria is motile. In that case, flagella will be there. Flagella will be there. Okay. So in this case, we can say main locomotory structure. Main locomotory structure, and means rest of the part will go in deep in the class, but here just keep in mind, so this flagella will help in what? Locomotion, or we can say it's a movement. It's a movement. Conjugation means here it's a transfer of genetic material from one bacteria to another bacteria. So these are the all structure that is related to what? Cell membrane. Related to the cell membrane. Now, once you enter into what? Enter into the cytoplasm. enter into the cytoplasm. So now I will go for another part, that is the structure which is present in what? Cytoplasm. So can we say that the first structure which is present in the cytoplasm, and that too, it's a useful usage of flagella is nothing but locomotion. So it's a locomotory structure. So it will help in going from one place to another place. Obviously, infinite bacteria can reproduce sexually. Obviously, it can't reproduce sexually. But it can mimic. It can mimic the sexual reproduction. But actually, it's not sexual reproduction. So it's a cytoplasm. In the cytoplasm, what are the structure we have seen? So the first structure is, let's say, ribosome. What type of ribosome? So here we can say 70S type of ribosome, and S stands for what? Sedimentary coefficient. Sedimentary coefficient, or we can say it's a Schwarzschild unit. Schwarzschild unit. It's nothing but a unit that will define what? That will define this, the size of that. The size of that. Now, what is the function of the ribosomes? So the main function of the ribosome is protein synthesis. So protein synthesis is nothing but the function of ribosomes. Okay. After that, what else the structure is present? The most important structure is its genetic material. So it's nothing but the genetic material. Can you tell me what type of genetic material? DNA or RNA or both? So genetic material, either it's a DNA or it's a RNA or it's a RNA. But in this case, DNA is a genetic material because virus is having the RNA. Virus is having the RNA. So here we can say DNA is what type of DNA? Double. It is having not one strand, it is having two strands, and also it looks like what? Also it looks like circular or linear. So here we can say double stranded circular DNA. Okay. Now next one is here we can say one more part is there that is related to the genetic material. I can say it is having the false nucleus. It is having the false nucleus. So that false nucleus is known as nucleoid, or I can say it's a genophore, or we can say it's a false nucleus. False nucleus, and that is nothing but its own chromosome itself. Okay, so yes, obviously our time is almost completed. So today, what are the team is playing IPL? So now I will just take few doubts, like if you are having any doubt. So is DNA replication continues? Yes, DNA replication, whenever one cell divided into two cells, that two cell will ask for what? That two cell will ask for the DNA, or we can say genetic material. So in that case, you have to replicate. If you don't have the copies of what? DNA, obviously the daughter cell will be very sad, like the parent is not giving what? Parent is not giving the DNA. So it's a duty of the parent, or we can say it's a parent bacterial cell, that it will replicate first so that it can distribute among the daughter cell. Bacteria reproduce by spores. Yeah, it can reproduce by spores. Yes, spores is there, no doubt. Flagella and fimbriae, what is the difference? So fimbriae will help to attach to the surface, but flagella will help in moving from one place to another place. What is the difference between the between mesosomes and mitochondria? So in both the cases, oxidation is there. You will get the energy. But obviously a structure is little bit different. One is having the advanced structure and other one is having what? It's a very simple structure. So mesosomes is very simple structure. Mesosome is very simple structure and mitochondria is very advanced structure. Difference between single and double. So obviously, like a single strand, like only one strand will be there. Only one strand, or we can say only one string will be there. So double strand means two string will be there. Two string will be there. Okay. Sedimentary coefficient. Sedimentary what? Sedimentation. It is going down. And how fast it is going down? So that based on that we can say it's a sedimentation coefficient. Mycoplasma have cell wall? No, mycoplasma is not having the cell wall. And that is kind of exceptional part. That is a kind of exceptional part. So I think I should pause my lecture here. So we'll meet you in the next class. So bye-bye everyone. Bye-bye, enjoy, good night. We'll meet you in the next class with the new topic, or we can say sometime, like we'll continue this topic itself.